Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled Reductions in Impulsivity in an Animal Model of Attention Deficit Hyperactivity Disorder, or ADHD. This webinar has been sponsored by PanLab, Colborne Instruments, and Harvard Bioscience, so a big thank you to them for helping to make this event possible. Joining us today, we're fortunate to have Dr. Scott Hall, an associate professor at the University of Toledo. His presentation will discuss assessments and treatments for the impulsive behavior commonly associated with ADHD. And without further delay, I would like to welcome Dr. Scott Hall. Scott, thanks so much for being here with us today to share your work, and the floor is yours whenever you're ready.
All right, thank you very much. First of all, I'd like to thank PanLab, Colborne, and Harvard Bioscience for organizing this, as well as Insight Scientific for running it. It's been a great experience, actually. I guess we're all getting used to these types of online presentations, but I think they may be something of the future as well, too. So today I'm talking about attention deficit hyperactivity disorder, and in particular, an animal model that we've been working on for quite a long time. It actually came out of work in drug addiction and sort of developed into a better model over time, or at least we realized that it was a better model over time. Now, first of all, when I talk about attention deficit hyperactivity disorder, I assume that most of us are familiar with that, but just to review, it is a disorder most commonly diagnosed in children. It is characterized by uninhibited responses, a lack of sustained attention, and hyperactivity. It generally has a childhood onset. Sometimes the symptoms abate as children grow older, and they're less severe in adulthood, but certainly adults also suffer from ADHD as well. There are a couple other interesting points, including the fact that there's a strong genetic component. This is not to say that it's by any means 100% genetic, and there are lots of environmental factors that also influence ADHD development. In addition, there's a lot of comorbidity of ADHD with other disorders, including addiction. And in particular, smoking rates are twice the population rates in individuals with ADHD. And there's also a very substantial sex bias. However, I've always been a little bit suspicious of how big that bias truly is. And one of the problems here is that the symptom profiles seem to be a little bit different in boys and girls. The boys tend to do things to draw attention to themselves, and the girls may simply have poor attention and do badly in school, so they often may be missed for that reason. Now, of course, the description I've given you is from the DSM-5, which really broke down ADHD into two categories. One in which the primary deficits were inattention, and one in which they were primarily hyperactivity, impulsivity, and then there's a combined version. However, I'm not sure that way of thinking about ADHD is a very good one, and I really am somewhat partial to the RDoC perspective here that really considers the different symptoms of ADHD to be separate. That is, that they may have different underlying causes that may require different treatments as well, too. So from the RDoC perspective, the areas of impairment that you would find in the DSM classification of ADHD would include in areas of vigilant attention, reward-related processing, reaction time variability, and so forth. It remains to be seen how well those RDoC descriptions actually match what we see in people. And we'll see how well that works over time. One of the problems here with trying to classify these sorts of disorders is that, generally speaking, when you get to the clinic, things tend to be based upon, diagnoses tend to be based upon what you see, what you observe. So you identify symptoms of inattention or hyperactivity or impulsivity by talking to patients or talking to their parents or teachers to identify the problems that they may have. Now, of course, this doesn't link the condition to any underlying biomarkers or biological function at all, which is really what RDoC seeks to do. However, it does not establish what those biomarkers would potentially be. So that's just a little bit of perspective on this. I would also add one more thing, is that when you look at the DSM criteria, there are several aspects of ADHD that I think are very important. And then when you talk to parents of children with ADHD, they think are very important, that aren't really encapsulated within the DSM criteria. And among these include deficits in decision making. Some of the things that parents fear the most in their children with ADHD is that they're going to harm themselves, not intentionally, but through poor decision making, which is kind of represented by the diagram here of the children jumping between roofs. But there may also be long-term changes in decision making that may relate to things like smoking or drug usage as well too, and may account for some of those comorbidities as well. Now, when you think of disorders of this type, one of the things to think of is aspects of the disorder that are less well treated than others. Very often we develop animal models by focusing upon particular causes or sometimes particular symptoms within a disorder. And when we develop drugs based upon those models, that means that we may treat those symptoms very well, but not others. And I'll return to that idea at the end here a little bit. So the model I'm going to talk about is what I would call an accidental model. That is, we didn't intend to develop an animal model of ADHD at all. And by we, I mean my colleagues, Chirosora and George Uhl, developed dopamine transporter knockout mice in the 1990s, and they did so to study addiction. They wanted to study the mechanisms underlying the actions of stimulant drugs. Now, when they did that, of course, one of their expectations was that by removing the dopamine transporter, that you would potentiate dopamine actions, at least within the synapse. And that is, in fact, what was seen. And based on that expectation, you would assume that there would be certain behavioral changes associated with that. The one that people would think of the most, of course, would be hyperactivity. And the figure in the upper left there shows one of the first diagrams from one of Chirosora's papers that shows pronounced hyperactivity in dopamine transporter knockout mice, in particular in the homozygous knockout mice. The heterozygous knockout mice did not seem to be affected. In addition, when you gave them cocaine, it didn't produce any more locomotor stimulation. Now, when you did the experiment differently, that is, you gave the cocaine not 3 hours after beginning the locomotor testing, but only after 30 minutes, you got a different pattern of effects. And this started to change people's thinking about what was going on here a little bit. So the figure on the right is from a Ganetinov paper from '98. And what you see there is in the first column, A, B, C, D, you see the wild-type mice. So very nice locomotor stimulant effects to a variety of drugs. The dopamine transporter knockout mice don't, but more than that, they show reductions in locomotor activity in response to methylphenidate and amphetamine. So this would appear to be the paradoxical locomotor reducing effects of stimulant drugs that people have noted for ADHD. Now, we can debate the nature of that substantially, and I will a little bit as we go on, but I'll return to that in a little bit. In this particular paper, they went on to try to suggest that the actions of these drugs were actually mediated by the serotonin system. And that's based on the drugs that you see in the last column there, I through L. However, I was always very suspicious of that for a variety of reasons. Most particularly this one, is that when you look at the L-tryptophan data in the lower right in particular, it's not that locomotor activity was reduced, it's that it was eliminated. So I was very suspicious that there was something else that might be going on here. And we subsequently did this paper where we showed that the dopamine transporter knockout mice were far more sensitive to the 5-HT behavioral syndrome. So we think that this was competing with locomotion. So it wasn't necessarily that you were reducing locomotion in the way that you usually expect, that this was a different type of effect. Now, we don't think that this was necessarily relevant to the actions of the stimulant drugs, though. but we think it accounts for this. Now, also in that paper, I just want to show you this to give you a little bit more of an idea of what we mean by locomotor hyperactivity here. Because what you might think is simply that the amount the animal moves goes up, which is true, but also the pattern of movement changes substantially. So the variety of measures here that you can get from the locomotor tracks that are represented in C there. In the first two, you can see that the mean meander and the turn angles both decrease. So what this really means is that the animal's movement patterns have changed. Instead of making a lot of small movements within an area and the movements being highly variable, they tend to be longer movements that are sustained over a long period of time. Basically, they tend to circle around the edges of the apparatus, and they do so for a very long period of time. Now, one of the things about this is that if you look at any animal's response, when you first put them into a box like this, that is their initial response. So partly what the DAT knockout mice are doing here is that they are persisting for a much longer period of time in their initial behavioral response. And we actually saw this across a variety of circumstances, not all of which I'm going to discuss today. But what I want to try and argue is that this ended up being an animal model of ADHD, and really ADHD in the sense of a DSM. So not that it showed some of the symptoms that are associated with ADHD that you would think of in terms of RDoC, and they are separate, but really it showed a cluster of symptoms that really does look like ADHD. And it shows a variety of changes at a biological level that are also similar to things that have been suggested for ADHD. So one of the things that first started to suggest this to us was this study. We did an in vivo microdialysis study where we really showed two things. So first of all, the basal levels of dopamine extracellularly were substantially increased. Oops, sorry. And you eliminated locomotor stimulant effects of cocaine. At the time, that's what we were interested in. But the interesting part of this study is that we actually also looked at the prefrontal cortex. And there you see a very different pattern of effects. So first of all, the basal levels of dopamine extracellularly are relatively unaffected. Now, if you look very closely with voltammetry, you can still see some differences. Just like with the heterozygous mice, you can see differences with voltammetry. But it was much less affected. And when you look with microdialysis in the prefrontal cortex, at basal levels, it was unaffected. When you look at the responses to cocaine, they're unaffected as well, too. So one of the ways to think about this is not just from the point of view of the striatum, where you have these changes in dopamine function there, this dopamine hyperfunction. But what has happened in these animals is there is an imbalance between cortical and striatal dopamine function. And this is something that is reminiscent of ADHD. Again. Now, are the behavioral changes we talked about, or even these types of changes in corticostriatal function, going to be highly specific to ADHD? I really didn't think so from the very beginning, because when you look across a broad range of what we would call frontostriatal disorders that include ADHD and schizophrenia and bipolar disorder, among others, you really see a substantial overlap of some of the symptoms, including some of the ones that aren't mentioned here that have to do with cognitive function. So it may be more that this imbalance between cortical and striatal dopamine function is a contributor to a variety of conditions, perhaps. I really didn't think that it would be unique to ADHD in the beginning. But as we go on, the evidence does kind of push us in that direction, I do have to say. So one of the next things we did was we were actually thinking more about schizophrenia than we were about ADHD. We were thinking that hyperdopamine function would lead to impairments in prepulse inhibition, which had been used as an animal model of schizophrenia. That is, by potentiating dopamine function in different ways, you would produce impairments in prepulse inhibition of startle that were similar to what you saw in schizophrenia. And when you gave antipsychotic medications, you would reverse those impairments. So if you're not familiar, prepulse inhibition of startle is a sensorimotor gating phenomenon. So if you have a very loud auditory stimulus, it produces a startle response, which you measure with the apparatus shown there on the left. The startle response itself is not often what people think. It's not jumping. It's actually lowering your center of gravity. And that's what's measured in the apparatus. However, if that pulse, if that auditory stimulus that produces a startle is preceded by a very short time by a prepulse, that is a stimulus that by itself is not startling, but precedes the pulse, it will reduce the startle response. So this is what prepulse inhibition is. And I say here it's a model of attention. It's really more of a preattentional process than a true attentional process, however, it developed into something a little bit more interesting in this model. Now, first of all, deficits in prepulse inhibition are by no means limited to schizophrenia. You actually see them in a variety of what we call frontostriatal disorders, including ADHD. So there have been a number of studies that show under certain conditions that there are deficits in prepulse inhibition in ADHD. So we did prepulse inhibition in the dopamine transporter knockout mice, and they were substantially impaired. Where it became interesting was when we gave them stimulant drugs that are used in the treatment of ADHD, like methylphenidate. In the wild type mice, the methylphenidate impaired their performance, whereas in the knockout mice, it improved their performance. So it normalized the sensorimotor gating deficits, which again are kind of a pre-attentional process. So you have, again, this sort of paradoxical effects of psychostimulants, where in the wild type animals, it's making them worse, and in the knockout animals, it's making them better. And that's somewhat reminiscent of how these drugs actually work in people as well, where in people who do not have ADHD symptoms, stimulant drugs tend to produce impairments, whereas in people with ADHD, they tend to produce improvements. There are questions there of dose, but we'll skip over that for the time being. Now, one of the big questions here is, how is methylphenidate really working to producing these beneficial effects in dopamine transporter knockout mice? So we looked at a variety of other drugs, including serotonergic drugs. But the most interesting effects that we found were with nisoxetine. So if you think of the effects of amphetamine, or methylphenidate, they're somewhat non-selective monoamine blockers. Nisoxetine is a selective net blocker, and it very effectively reversed the deficits in the dopamine transporter knockouts in PPI, while having no effect in the wild type animals as well. So presumably the impairments that were being produced in the wild type animals had a different mechanism from the improvements that were seen in the dopamine transporter knockout animals. So the next question we asked was, where does this effect happen? So in the next paper, what we did was we injected the nisoxetine directly into the brain, into two regions that we thought might be relevant to this behavior, the prefrontal cortex and the nucleus accumbens. When the nisoxetine was injected into the prefrontal cortex, it dramatically improved their prepulse inhibition. So it reversed the deficits that you saw in the DAT knockouts, had no effect in the wild type. When you injected the same drug into the nucleus accumbens, it had no effect in either genotype. So you did not see improvements in the dopamine transporter knockout mice. So this is saying that really it's saying that the methylphenidate and the amphetamine are probably acting through the norepinephrine transporter to produce beneficial effects in this model, and they are doing so in the prefrontal cortex. So next we tried to look for what circuits the nisoxetine might be working through. So we began by looking with CFOS activation. So we looked in a variety of brain structures that were interconnected to the nucleus accumbens, and a few structures that were not. And it was very interesting that all of the structures that had projections to the nucleus accumbens shell and the nucleus accumbens shell itself had very strong activation of CFOS by isoxetine. So this included the medial prefrontal cortex, the basolateral amygdala, and the ventral hippocampus. But when you looked in other regions, you did not see this. So you did not see any activation in the NAC core or in the dorsal hippocampus, for instance. And then we did double labeling. So the isoxetine activated cells in the prefrontal cortex were glutamatergic. The ones that were activated in the nucleus accumbens were GABAergic. Now, I'll point out one little thing here, which is kind of interesting, which I think guides some of our thinking. This is just one small example of this. But the basal level of CFOS activation in the prefrontal cortex was low. It was actually reduced by almost 50% in the DAT knockout animal. So there seem to be changes at baseline in the dopamine transporter knockouts in the activity of prefrontal cortical neurons. And again, these would be glutamatergic projection neurons as well. So we then did triple labeling to try and see where those neurons were projecting. And we used Florigold injections into the nucleus accumbens or into the VTA. And we got very strong triple labeling of these neurons. So the isoxetine activated neurons appeared to be glutamatergic, and they appeared to primarily project to the nucleus accumbens as well. Now, when we looked at some other ways at those prefrontal cortical neurons, we saw changes. So one of the things we looked at those pyramidal neurons. There's one shown in green there on the right, along with a close-up of some of the dendritic spines. And we did spine counts, and we found that there were reductions. In particular, there were reductions the farther away from the soma you got as well, too, which is where most of the glutamatergic contexts really are. So there were substantial morphological changes in the prefrontal cortex glutamate neurons. Now, so far, in terms of all of the behavior that I've talked about, I haven't really talked about anything that is terribly relevant to cognition. And if we really want to treat ADHD, we need to think about the attentional and cognitive deficits. Now, we did do this task relatively early on, one that I am rather fond of and I think is very meaningful, but I'm not certain how many people actually agree with me about that. So this is something called the cliff avoidance reaction. Now, one of the things that I find very interesting about this is that it shows how completely different the dopamine transporter knockout behavior is from the wild type. So this is just a series of stills. This is actually published originally in this paper. And forget that, 2013, so a few years ago. And if you look at the animal on top there, the wild type, it barely goes near the edges. It keeps its body very low, flattened against the platform. Basically, this is a small platform. It's about 15 centimeters in diameter. It's elevated about 40 centimeters off the floor. So there's a visual cliff as the animal goes near the edge. It has a highly beveled edge as well. So the wild type animals barely go near the edge, and importantly, they never fall off, whereas the DAT knockouts would consistently fall off. But also, when you look at the DAT knockout, its behavior is just intrinsically different. It doesn't seem to be bothered or concerned about its circumstance. So it's much more elevated on its feet as it walks around. It goes very quickly up to the edge. It wanders around. It peers over. Eventually, it tries to climb underneath and falls. And what you actually see when you test a lot of animals here is that DAT knockouts fall a lot. In some more recent work, my graduate student also showed not only do they fall, because in this experiment, we only let them fall once, and then we ended the experiment. But he actually put them back on the platform, and they would fall repeatedly as well. Now, when we first submitted this paper, I got into a little bit of back and forth with the reviewers because they were arguing that this meant that they were impulsive. They thought of this failure as a deficit of motor impulsivity, which I didn't think was true. And the reason for that has to do with that diagram in the middle. That is, the animals were not falling as soon as we placed them on the platform. They thought about it for a long time and still did something that was intrinsically dangerous. They fell, many times their body length to the floor. We had a nice little padded thing for them to fall into, but still, they did something that was intrinsically dangerous. They didn't know how far it was. They didn't know whether they might hurt themselves. They were doing something that I think is very similar to that first picture I showed you of the children jumping between the roofs. And this is something that children are more likely to do than adults, but children with ADHD even more so. Now, again, another interesting thing here is we gave them a drug, which somehow the drug isn't labeled here, but we gave them a drug that is useful in the treatment of ADHD, which in this case was methylphenidate, and it again normalized the deficit. It had a slight effect, slight impairment in the wild type animals. That was not significant, but the improvement in the DAT knockouts was very significant here. So here's a slightly more recent study. So far, when I've talked about the DAT knockout model of ADHD, I've spoken about homozygous knockout mice. And I haven't really mentioned the heterozygous knockout mice. In some studies, we simply did not look at them. In some studies from other investigators, they also did not look at the heterozygous mice. There's often been a tendency to ignore heterozygous mice. Personally, I think they're far more interesting often than the homozygous knockout mice for a simple reason, is that the range of variation in gene expression that you have in heterozygous knockout mice is far more common to observe in humans than a complete knockout. You do not see complete knockouts of the dopamine transporter gene in humans. It is incredibly rare and very deleterious. So it has been observed a few times, but it is by no means something common to see in the population that might really be relevant to ADHD. So saying it may still be an interesting model. But we looked in the heterozygous mice more recently. This is unpublished data now. And we found a variety of things. First of all, we looked in males and females. And one of the things we found is that there were impairments, CAR deficits, in heterozygous mice, in particular male mice, but also female mice. These were smaller than what we observed in the homozygous mice. But the most interesting thing here is I'm not showing those comparisons here, but the males were far more affected than the females. And this is something that we saw, I'm not going to go into too much today, but we definitely saw large sex differences in this study. And we looked across a variety of behaviors. So that the presentation of behavioral deficits in DAT knockout mice was different in males and females. But here, what I really want to point out is that when you gave these animals atomoxetine, you again saw in the male wild-type mice an impairment in CAR. In the heterozygous male mice, the deficit was improved, as it was in the homozygous mice, and you saw the same thing in females as well, too. The atomoxetine improved the impairments that you saw in the CAR test. And it's difficult to say what that is. A lot of people kind of think of this as a test of impulsivity. I really don't think that's true. I really think that this is really reflecting risk assessment more than anything else. And I'm happy to debate that with anybody who has a different opinion. So based on some of the data I've shown and some other data that I have not, we sort of developed this circuit model of how we felt a lot of the drugs that were tested were working. And basically, we thought that the stimulant drugs were working through the norepinephrine transporter to potentiate dopamine function, but perhaps norepinephrine function as well. We're not certain of that too. But the reason that in the homozygous mice, the levels of dopamine were not affected is that there's very low levels of dopamine transporter in the medial prefrontal cortex. You simply don't find very much of it within the dopamine synapses, which means that the primary mechanism for eliminating dopamine from the synapses in the prefrontal cortex is through the norepinephrine transporter, which is often at a slightly greater distance from the dopamine synapses as well, of course. So the dynamics of dopamine is fundamentally different to begin with, but a consequence of this is that when you remove the dopamine transporter in the medial prefrontal cortex, it doesn't have much of a consequence because they're pretty low levels there to begin with. Now, we also showed that there were effects of nicotine on the deficits and DAT knockouts, and we showed that those effects of nicotine were also dependent upon an interaction with serotonin neurons. And as we worked through the circuitry, we proposed that certain serotonergic drugs might be useful in treating some of the deficits that we saw in our DAT knockouts here, and by implication, potentially in ADHD, if this is really a model of ADHD. So at this point, we've shown a variety of symptoms that are related to ADHD that include hyperactivity, maybe not attentional deficits at this point, but at least preattentional deficits, impairments in executive function in a couple different tasks. I would say those deficits in the CAR test are really reflecting risk assessment. But we haven't done any more complex tasks. We've shown some changes in cellular and anatomical function that seem to implicate changes not just in dopamine function, but changes in corticostriatal projections. So collectively, this data, which includes for many tests, reversal of the impairments by drugs that treat ADHD, the model seems to have a high degree of validity, so both face validity and predictive validity, but also some degree of construct validity. That's always difficult to assess because we don't fully understand what causes ADHD in humans, of course. Now, when you look at what I've done so far, or what I've summarized so far, there are obviously certain shortcomings. And a few years ago, when I moved to the University of Toledo, we sought to try to improve upon that state of knowledge. So in particular, the main shortcomings of the evidence had to do with the fact that we had not really explored any more complex task that was very directly relevant to attention or impulsive behavior. So these sorts of tasks would include the 5-choice civil reaction time task, the continuous performance task, go-no-go tasks, and others that are similar. In addition, the DAT knockout, the complete DAT knockout, is not really observed in humans. So the construct validity of the model is problematic at that point. Now, still, it might produce a state that is similar in some respects to ADHD. But the DAT knockout itself is certainly something that is not observed in individuals with ADHD. You might see reductions in dopamine transporter expression, although even that is somewhat debated and does not seem to be found in ADHD, oddly enough. Now, we also, along the way, I haven't discussed this story very much so far, but we hypothesized that 5-HT1B antagonists would reverse some of the deficits in the DAT knockouts. But we hadn't shown that in very many ways so far. And in particular, when we began this series of studies, we'd really only looked at locomotor hyperactivity. Now, 5-HT1B antagonists did reduce locomotor hyperactivity, but in order to be at all confident that they were doing something meaningful in the model, we would want to look in some other circumstances. So we decided to look at really a series of tasks using these 5-choice boxes that are shown on the left there. So these are the PanLab Harvard Bioscience 5-choice boxes. They have 5 nosebook holes on one side of the apparatus, which are represented by the light bulbs there. And basically, the animal has to wait for the light to go off in the nosebook hole and pick the right one. So there are 2 parts to that. The animal has to wait and has to respond appropriately. And you can change the conditions that are involved in waiting and responding. One difference from the apparatus shown there on the left, I should have gotten a picture of our actual box, but we actually used the liquid dispensers. And the reinforcer that we were using is strawberry milkshake, which the animals loved, and it was very easy to train them on. So not going to show you any figures for a little bit. I'm just going to describe the next part. So to begin with, we did the 5-choice serial reaction time task, and we began by looking at homozygous DAT knockouts who were absolutely hopeless. They could not learn even the most basic responding in this apparatus. Now, this was despite the fact that we had done a previous study in which we had an operant-based food task and an operant-based cocaine self-administration task, in which they were perfectly fine, at least under basal conditions. And for the food conditions, they were always fine. They even did a progressive ratio. So in that task, they were fine. But in that task, the food came out right next to the lever. In this box, the reinforcer, in our case, strawberry milkshake, is dispensed at the back of the apparatus away from the lights. So what happened with the homozygous dopamine transporter knockout mice is as soon as they figured out the food was coming from the back, they turned around and stared at it forever and never stopped. So they never noticed the lights going off and wouldn't respond. And they couldn't even learn the most basic version of the task in which all the lights come at once. They stay on for like 30 seconds and all the animal has to do is nose poke. They couldn't even learn that. So it was hopeless to take them to anything where we could look at impulsivity or attention. It was a very interesting circumstance. My graduate student actually ended up doing a whole bunch of things to try and teach them. He tried to give them drugs during the training to try and improve their performance, all to no avail. So we actually moved on to the heterozygous DAT KO mice, which we're actually more interested in a variety of respects. It might be more like what's going on in humans, and the effects may be more subtle than what we saw in the homozygous knockout mice. Now, when you look at other things, the heterozygous knockouts show very subtle deficits in most of the other tasks that we've seen. You can get a locomotor hyperactivity, but it's very small. You can get small PPI deficits, small deficits in CAR, but they can be significant. And you can reverse them with drugs that treat ADHD in those tasks. So we were hopeful that they would show more pronounced effects here. So we began by doing the 5-choice serial reaction time task, and they learned it perfectly fine and showed no deficits, at least under the conditions that they were studied. Now, that's something that's very important here. So it's not just a matter of doing any of these tasks, whether it's the 5-choice serial reaction time task or the continuous performance task. A lot of it has to do with the specific conditions that you test under. And that's shown in some of our results. So the heterozygous mice did not show any deficits in the 5-choice serial reaction time task. So we moved on to the continuous performance task, which includes a go, no-go component. So that go, no-go component is represented on the right, where basically the top part is identical to the 5-choice task, where one of the lights is lit on each trial, and they have to respond appropriately. This includes waiting until the light comes on to respond. If they respond before the light comes on, that's an impulsive error. However, 20% of the time, all of the lights would go on. And in that case, the animal had to withhold responding. So when we did the task under this condition, first of all, we also had very varied conditions on test days. And this was actually important to identifying some of the deficits. So we had different stimulus durations, and we had different intertrial intervals. And in particular, the intertrial intervals really brought out some of the deficits. Now, you can see here that the percent correct and the percent emissions was changing substantially with things like stimulus duration. So they had higher performance when the stimulus duration was longer. There were some effects here of amphetamine, but they weren't in any way specific to the DAT knockout. So we didn't really see any differences in attention under these conditions. Now, I'm not confident, or I suspect, that we can bring out those differences. We just haven't hit on the right conditions. But we ended up finding something else that's more interesting that I'll get to in a minute. So we actually were testing a couple different drugs. As I showed here, we tested amphetamine. We also tested the 5-HT1B antagonist. That's SB224289. So again, here you see just sort of the basic kind of pattern of effects you get with changing the ITI and the stimulus duration. It got interesting when we looked at premature responses. So in the DAT knockouts, at the long intertrial intervals, what you found was a much higher rate of premature responding. So they are having difficulty with holding the responses during the challenging conditions. So when the intertrial interval was short, it was not much of a problem. But when you extended the intertrial interval, then they started to have trouble. Now, in addition, when we gave them either the amphetamine or the SB 224289, it reduced that impulsive behavior. So now, for the first time in our model, and now our model is extended to this less severe form in the heterozygous knockout mice, we have evidence of impulsive behavior, and we have evidence that drugs that treat ADHD reduce that impulsive behavior. And we also have evidence that our potentially novel approach to treating ADHD also produces a reduction in impulsive behavior. Now, one of the things you might also say here is that this measure of impulsive behavior is not necessarily specific to ADHD. It may be that impulsive behavior that's associated with other psychiatric conditions might also be treated by these drugs too, which I suspect may very well be true. So we moved on to some other tasks to try to assess what sort of cognitive changes might be going on in a mice. And one of the things we did was the Iowa gambling task. So in this case, we used 4 of the holes. It was a 3-day task. So on the first day, they were forced to respond to each hole 20 times. And they learned that the first 2 holes would give out 1 reinforcer, whereas the second 2 holes, 3 and 4, would give out 2 reinforcers. So then we would look on the second day at reward discrimination. In addition to the amount of reward, the probability of reward was different across the holes as well. So the probability of receiving a reinforcer was 50% on holes 1 and 3 and 75% on holes 2 and 4. So this should, again, sort of bias their responding, particularly in the long term. On the third day, we added a punishment component. So basically, the holes 3 and 4 were much more highly punished. So what this means is that compared to day 2, their behavior should shift on day 3 away from those holes that have the more severe punishment. And the punishment here is just the house lights turn off and a new trial is not started for a period of time. So the punishment probabilities differed a little bit, as well as the duration of the punishment. So you can see it's far more severe for holes 3 and 4, 222 seconds or 444. So this is the behavior that's represented here. So you can see here that they very quickly figure out which holes give more reinforcer. You can see that in the upper left there. In addition, there's a rather interesting thing in that they definitely had a preference for the outer holes as well. So there were other biases that sort of overrode how much reinforcer they were getting. And then when you moved from day 2 to day 3 results are represented in the lower left. They generally shifted their responding to the holes that were not being punished, which is somewhat natural. And there didn't seem to be any differences here between the heterozygous DAT knockout mice and the wild-type mice. However, my graduate student, Yasser Saber, did something sort of clever. He didn't just look at the overall performance, but he looked at the shift in their performance. And that's what's represented in that third graph there, is their choice preferences adjustment after the reward devaluation. And you can see very clearly there that the wild-type animals shifted their responding more towards the beneficial holes and away from the less beneficial holes, much more than the heterozygous animals do. And there's a sum of that for each of the 2 holes in the graph just below that. Now, in addition, this was, again, a trial-by-trial kind of test. We also had premature responding that was increased in the DAT knockout. So it's the second type of task where we've gotten this type of increased premature responding or impulsive behavior in the DAT knockouts. So, conclusions regarding the dopamine transporter heterozygous mice as an animal model of ADHD. They don't show the extreme behavioral manifestations of the homozygous mice, but they still show deficits that are relevant to ADHD, and in particular, some of these more complex behaviors. And this includes impulsive behavior. And again, just like some of the simpler behaviors, the impulsive behavior can be reduced by drugs that are effective treatments for ADHD, as well as our putative drug here, which is the 5-HT1B antagonist. They show a variety of changes here that I think are really reflective, not just of impulsivity, but changes in executive function overall. And this includes shifting their behavior between choices in day 2 to 3 of the IGT. Now, in terms of what deficit really underlies that, is it perseveration or poor inhibitory responding? It's a little bit difficult to say just based upon that data, but it definitely shows a relatively specific type of deficit, and one that it could be assessed pharmacologically as well too, although we have not done that yet. So these are, I presented a lot of background data at the beginning of this talk, so it's a long series of studies that was done when I used to be at the National Institute of Drug Abuse, where I worked with George Uhl and Ichiro Sora for a very long period of time, and whom I continue to collaborate with. And many of Ichiro Sora's students were responsible for a lot of that work. The last work that I showed you with all of the operant tasks was done by Yasser Saber, who just recently got his PhD and became an assistant professor back in Iraq, where he was from, at Nineveh University. He was also assisted by several undergraduate research assistants during that. And of course, I'd like to acknowledge the support for the work. We've had some support from NIDA, as well as from the University of Toledo. Thank you for your attention.
Wow, thank you, Scott, for that amazing presentation. It was great to hear about your work. So
Can you hear me?
Yes, we can. Okay, so we're gonna kick off the Q&A here with the first question from Elizabeth. She is asking, how long does it take to train mice for the 5-choice test?
Thanks, Elizabeth, for asking that. I sort of skipped over the details of that. Any of you who are familiar with the 5-choice task knows that it takes a very long period of time to train the animals, often 5 to 6 weeks. However, my graduate students, based upon a number of recent publications, tried to accelerate that training. So basically, he did mass trials for essentially as long as the animals would stay motivated. So basically 100 trials at a time. But he did that 3 times a day. So he was able to reduce the training phase of the 5-choice reaction time task down to about a week. So it was maybe a week to 10 days in the animals that were a little bit slow. And this included both the heterozygous knockout mice and the wild type mice. And there didn't seem to be a difference in the time of the training between them. And this was actually something that was important to us. One of the things I've ignored here too is that this is a disorder that we commonly treat in adolescents, in children. So we really should not be studying this in adult mice at all. All the mice I was talking about were adults. We should be studying in adolescents. But when we started all of this, the training would take 5 to 6 weeks. So now, once he was able to get it down to a week, we were able to actually do an assessment in adolescents. And I didn't report that data here, but what it appears is that in the adolescents, they show greater attentional deficits than the adults do, which is interesting because that's also a pattern you see in humans where some of the deficits tend to get better a little bit as humans get older.
Great. And that's something that's going to be really exciting once you guys get that training down to a week, which you have. I think those studies will become really a little bit more elucidating in the impulsive behavior. So that's really fantastic. Okay, so moving on to our next question. This question is from Frank. Do the heterozygous knockouts show the same changes in circuitry as the homozygous knockouts?
So, yeah, good question. We do not really know. The one study that really looked extensively at that, actually, all of the studies that looked at circuitry at all. So there were several others I did not mention, but the RMA study and the Lee study, all of those only looked at the homozygous mice. So we don't really know what's going on in the heterozygous mice, which is interesting. If you look with voltammetry at dopamine release, you see very subtle changes. So that was work of Sarah Jones from back in the 90s. So the heterozygous mice definitely show some effects upon dopamine, but whether that extends to some of the broader changes we see in the homozygous mice, I can't really say.
Okay. Our next question is from Stephanie. She is asking, do you think that cliff avoidance reaction tasks could be a model of reflection impulsivity rather than motor impulsivity?
Maybe. But I don't think so. So motor impulsivity, the idea is you have kind of a prepared response. The go no go is like very characteristic of that, right? You have the inherent bias towards responding, and because of the bias, there's the tendency to go. With reflection impulsivity, the idea overall there is again that you act before you carefully consider the circumstance. And I think of the DAT knockouts here that it's some other type of executive function because I think they are carefully considering the circumstance. They're just weighing the potential outcomes differently, either their probabilities of occurring or how bad different consequences could be. And part of the way I think about this is through ADHD patients that I've seen. So, like a few years ago, we were up at camp, which is what people in Michigan call their kind of summer homes. And it was on a lake, and we were out kayaking. And my son had brought along a friend of his who had ADHD, and he was basically taking a vacation for the week from his ADHD medication, which meant that his symptoms got worse. And we were out kayaking, and I saw him paddling furiously in front of this boat. So it was a speedboat going about 30 or 40 miles per hour. And I yelled at him to stop. He was absolutely convinced that he could go fast enough to beat the speedboat. And this is kind of what I think happens in a lot of children with ADHD. It's that they're weighing the potential outcomes differently. So I don't think it's necessarily reflection impulsivity in the way that we usually think of that.
Okay, that makes sense. We have another question here from Kermit. Do the DAT knockout mice have normal cannabinoid receptor expression, like protein and mRNA levels?
Oh, no, we do not. No, sorry, we do not know. That is definitely something we have not looked at all. And to be honest, it's not something I considered looking at. It's an interesting question. My friend Emmanuel, who I was viewing this earlier, I'm sure would scold me for failing to think about that. But no, that would be very interesting to look at and relatively easy too.
Well, it's funny that you mentioned Emmanuel because he has put in a question here, and so I'll address that one now. He says, with FDA approval of CBD for epilepsy, is there a role for the endocannabinoid system in ADHD? And if so, what is the effect of cannabinoids in the DAT knockout mice?
We're getting into all those questions where it ends and I don't know. Hopefully I can say something a little bit more interesting. I don't know. There's an interesting point there. When I think of nicotine and ADHD, I think of self-treatment, particularly for some of the cognitive effects of ADHD. There's definitely a higher usage of cannabinoids among children with ADHD, I'm not convinced that it makes them better. So that may be more a matter of their decision making or their impulsive behavior. It makes them more likely to use drugs during adolescence. And also not thinking about long-term adverse outcomes. But I would not want to exclude the possibility that some cannabinoid use among children with ADHD is self-medication. And maybe it's self-medication for different symptoms from some of the other drugs. So when I think of the nicotine, I definitely think of cognitive symptoms. It may be some of the other symptoms that they might use cannabinoids for. So I wouldn't want to exclude that possibility.
Okay. Thanks for that answer. Before we move on to our final question, because we are running a little bit tight on time here, you'll notice that a survey has popped up on your screen. So for everyone that's still here, please participate in that survey. We'd love to hear from you. And I will now take our last question. So this last question is from Edward. He's asking, do you expect the PPI deficits to be related to PFC dysfunction as opposed to early sensory areas? And if so, how?
I don't think we have enough evidence to say where the effect is mediated. So certainly modulation of that corticostriatal circuit or the striatopalatal output circuit has been shown to affect PPI. So there's a descending circuit that runs through all of that down to some of those lower circuits. So I could certainly see the effects being mediated at a higher level, but I don't think I have enough evidence to say one way or the other where that effect might be mediated. Yeah, we'd have, well, I mean, we do have the evidence. Sorry, I've been talking a little bit too long. The nisoxidine in the prefrontal cortex did have an effect. So, I mean, that is showing that the higher level part of the circuit is modulating the lower levels. So that doesn't rule out some involvement of other structures, of course, but at least that area is involved.